We'll get going, everybody. So it's my pleasure today to introduce you to our colloquium speaker, Professor Boris Shraiman from KITP and UCSB. Boris received his PhD in physics from Harvard University, working with Paul Martin, and then went on to do a postdoctoral fellowship at University of Chicago and Bell Labs. He was then a member of the Department of Theoretical Physics at Bell Labs for about 16 years, and then did a short stint at Rutgers University before moving to his current position at Santa Barbara, where he's now the Susan F. Gurley Professor of Theoretical Physics and Biology and a permanent member of the Kavli Institute for Theoretical Physics. So Boris's work has spanned a really diverse range of topics, from the physics of quantum materials to the study of turbulent flows and how intelligent agents and animals might go about navigating through them. I first got to know Boris through his work in evolutionary dynamics when I was a graduate student. Among his many contributions in this area, he and one of his former postdocs, Richard Nair, derived some really beautiful results showing how you could predict which influenza strains are likely to take over the population in the next year solely by looking at the shapes of their genealogical trees. So really great stuff. Boris has also spent a bunch of time thinking about the interplay between mechanics and morphogenesis, which he'll be telling us a little bit more about today. And in recognition of his work, he's been the recipient of a number of prestigious awards, including the American Physical Society Fellow and a member of the National Academy of Sciences. And it's a real pleasure to have him here to deliver our colloquium today. So with that, take it away, Boris.
Well, thank you so much for inviting me. Thank you for coming. My pleasure and honor to tell you a little bit about physics in, well, let's say physics and biology. So morphogenesis is the process by which plants and animals generate their physical shape. So, for example, my arm here is a product of a morphogenetic process. And I will try to convince you that well, you know, more broadly physics in biology, and a relevant question in the physics department, also in biology department, what does physics have to contribute to biology in the first place? And I would roughly, okay, next technical difficulty is, can you see that little, okay. Let me split this into several bullet points. One sort of most obvious contribution is what I would call biophysics sensu stricto. So actual physical phenomena encountered in biology. Another bullet point is the physics concepts and methods that we use to make biology more quantitative. And last but not least is thinking for physicists so physicists thinking about biology is an opportunity to actually enlarge the domain of physics, discover new questions, new paradigms in physics. And perhaps last but not least is perhaps zero in on fundamental questions that biology has not asked yet. So let me give you an example of recent advance of the sensu stricto. We all learned about intracellular organelles in high school. More recently, it was discovered that a lot of intracellular processes, so biochemical processes, happen in membraneless organelles. Basically, liquid phase-separated droplets that enrich certain enzymes and concentrate certain biochemical processes. And remarkably, this was only discovered about 15 years ago by Cliff Brangwyn and Tom Hyman. and they got the breakthrough prize for this discovery this year. In a very different vein, let me give you an illustration of what you might call an off-label use of physics concepts. If you look closely at the fly wing, you will see a nicely combed array of little bristles. And if you zoom in, you will see that each cell has a little actin bundle. And as in this picture, they're very nicely ordered. But so there is a factor, if you like, polarity associated with each and every cell in epithelial tissue. And then there are some mutants where cells still grow their little vectors, but now they're disordered, right? So in this audience, the analogy with ferromagnetism is immediate and inescapable. But there is no magnetism involved in this phenomenon, right? So ideas are useful. Now, Darcy Thompson, 100 and some years ago, famously thought that morphogenesis, the description of shapes and processes that develop the shapes, belongs, as a physics problem and should be studied by physicists. Of course, he was thinking about these problems way beyond the advent of molecular biology and genetics. And we learned so much about development since Darcy Thompson. But so, for example, now we know exactly which gene, if expressed in, so ectopically out of place in fly's head, will turn an antenna into a leg, right? So just a single gene flips the switch and switches the antenna growth program into a leg growth program, right? And we can understand this genetics, we can know the gene, but we still have no idea of what is it that the actual growth program that gives rise to the shape of an antenna in one case and the shape of a leg in another case. So how is shape actually encoded in the process of development? So wouldn't it be nice now to revisit Darcy Thompson's intellectual agenda with the power of 21st century biology? We have some examples now where we understand a little bit more about this link between genetics and geometry. For example, there is one particular gene that is expressed in plant leaves, and its expression controls the shape of the leaf. And basically what's happening is, it turns out this gene controls growth. So its excess, let's say, in the center of the leaf leads to overgrowth in the center. And as a result, right, the shape of a flat disc leaf would cup out, right, to make more area for this extra proliferation and growth of cells. If instead you get more growth on the periphery, right, the shape has to form negative, surface has to form negative curvature, basically, to make more perimeter for the area that it's got, right? So, simple differential geometry ideas. And this inspiration from morphogenesis has been transplanted into the practice of sort of bi-inspired manufacturing here. In this particular case, one can sort of pre-program little gel discs so that once they're cured at high temperature, then they change shape from flat to curved in various ways. So pretty obvious. But the amusing thing is the plants somehow know differential geometry, and we can think a little bit more about this process. So when I was talking about differential growth, I was talking specifically about isotropic growth, right? And Gauss tells us that 2D surfaces embedded in 3D are conformally flat. So just by non-uniform isotropic growth, we can encode pretty much any shape we want. Well, curiously, plants, for example, do that, but growth in general is not isotropic, right? So now if the growth is anisotropic, instead of sort of one number to characterize growth tensor, we have three numbers to characterize growth tensor, right? So the extent of anisotropy and local direction in addition to expansion. And you may ask, well, who ordered that? Why would nature be doing with three parameters what can be done with one? And we can start thinking about it in some very general terms. Dylan Sislow, a graduate student at UCSB, thought about that. And for example, can show that by allowing anisotropic growth, one can achieve the same final shape with more uniform prescription of growth, if that's a good thing. So we can dream of some general principles, but the problem is that we can't really seriously think about optimization without understanding the constraints under which this optimization is to operate. So we have to understand, think more about mechanisms, the biological mechanisms that are constraining the process of morphogenesis. And I had this conversation with Surya this morning, and in some other context he said that when we don't understand some difficult problem, it means that we typically don't understand the simple problem. So let's move to a somewhat simpler problem that we also don't understand. Clearly, these three-dimensional shapes are a product of ultimately mechanical aspect of development, right? The physical shapes. So if we want to understand the mechanism, we also need to understand mechanics of tissues. And differential growth is not the only aspect of the mechanical aspect in morphogenesis. So let me change gears a little bit and now dive into much more detail into a story about early development of fruit fly embryo. So we're looking at, so this ellipsoid is, so catches fly embryo at the stage where it has generated several thousand nuclei, which are lining up the surface of ellipsoidal egg. And right, of course, initially, all these cells, all the nuclei are completely identical. The genes are all the same, but the first vestige of spatial structure manifests itself through expression of genes, right? All the nuclei have the same genes, but they become expressed differently. So red here shows you nuclei. Yellow is visualization of one particular gene being expressed. And now let me play a movie, and you will see first the formation of the stripes of gene expression, and then the next thing that's going to happen is this very dramatic rearrangement of nuclei on the surface. So kind of a massive flow of epithelial tissue on the surface of the embryo, and during this process, in the very beginning, I guess there's a rule that one has to play everything twice, but okay, maybe I won't. So instead, I'm going to play. I'm sorry. Here. I'm going to play this movie again, but this time with a different type of imaging, where we look not at the nuclei, but at cell interfaces. And another nice trick that Sebastian Streichhahn at UCSB, who has generated all this data, what he likes is unroll so the surface of the ellipsoid onto a square here. This is kind of Mercator projection of the embryo. And now I'll try to play it. Okay, come on. Okay, sorry, I'm having trouble with my visuals. Okay, I can't play it, but if I could play it, you would again see massive flow of cells on the surface. But instead of showing you the movie, I'll show you Sebastian's PIV analysis of velocity. So you're already seeing sort of the vectorial flow. And so topologically, the flow is sort of pretty simple. There are two hyperbolic points. So one on the ventral side, so the belly button, and then symmetrically in the middle of the back. And of course, topologically, this is a sphere. So if you have two hyperbolic points, you have to have four elliptic points, and that's a flow. And Ben mentioned that I have some background in hydrodynamics. So at this stage, I said, well, let's do this tissue hydrodynamics. Let's do sort of fluid mechanics of fruit flies. The only question is, so we're clearly dealing with a mechanical process. But what is the right description of tissue mechanics? Is it actually fluid or solid? It is very different from continuum mechanics that we learn from Landau and Lifshitz, in the sense that tissue is not deformed relative to a fixed reference. It is remodeled from the inside, actively. Right? And that's what we need to understand. And not only it is just remodeled from the inside, it is remodeled in some controlled fashion, right? There is genetic control and there is feedback. So that's what we need to understand. Okay, now let's pretend that we can peek inside this monolayer of cells and ask, what would we see inside cells if we could see inside cells? Well, there's a lot of stuff inside cells. We are focused on cytoskeleton, on the stuff that is directly relevant for generation of mechanical properties of cells and generation of tensions and forces. So the relevant structure for us is the actin-myosin cortex. So I remind you that actin is basically a structural polymer, and myosins are molecular motors that cross-link actins and can generate tension and contract these filaments. These cortices form kind of two-dimensional belts at sort of apical, so near the top surface of cells, right? So, interestingly, these cortices of individual cells are then cross-linked, so riveted together across cell membranes, and basically form a transcellular mechanical network. So that's the structure. So incidentally, I really like to encourage you to interrupt me and so start asking questions, because I get a little bit lost with the audience not asking questions. So please help me out to ask questions. Well, so at this moment...
can you go back?
Good, thank you, Daniel. You can always count on you. Yeah, how far?
That was my question. Can you go back? No. the catenyls are passive and the energy is just going in the myosins, or is there...
Passive. Coherence are passive. Okay. Well, so I introduced you to 3 molecular players. I assure you there are hundreds more. So an immediate question is, once we start on this road, just how far do we want to travel? Do we want to incorporate everything known to every biologist on the planet that could potentially be relevant? Or do we say that we're going to build sort of a minimal model and decide, hopefully judiciously, what minimum is? So, rather than making complex models, we're going to try making very simple models. And in fact, this model with 3 types of molecules is already a bit too complex for yours truly. So, I'm going to simplify it and reduce it to sort of a simple geometrical structure, right? Polygonal tiling of the plane, characterized by these positions of vertices. And we're going to equip the structure with some tensions that live in the edges and some pressures that live in, sort of 2D pressures. And we're going to start by thinking that these edges are basically representations of our actin-myosin filaments, and each filament has some reference length and is a little Hookean spring relative to that reference length. And the first thing that the tissue does is, so fast dynamics is to establish mechanical equilibrium where tensions balance. And going forward, I'm just going to assume that we can forget about pressure differentials, and pressure in the tissue is just there to, right, so the Lagrange multiplier on the area, so to prevent the collapse of the area under tension. So that's our fastest process. And then our cell array becomes kind of a tension net, and there are 3 tensions impinging in every vertex. They have to add up to 0 because we assume rapid equilibration. That immediately means that these tensions have to triangulate a plane. So that's different space, tension space. There is this sort of rigid triangulation that is dual to the polygonal tiling of the plane by cells. So if you do that, you notice that the existence of this dual triangulation implies a constraint on cell shapes. It's basically, it follows from a law of sines, and it's a statement that ratios of sines of certain well-defined angles, right, product around the plaquette has to be equal to 1. Right, so this is pure geometry and high school geometry at that. And it fell into our lap as an immediate consequence of force balance. Right? We didn't need to know anything about any molecules. Right? Just the assumption of the force balance, and this geometric fact falls into your lap, and you can immediately go back to the data and check it. Do these nice polygonal cells that you get from your experimentalist friends, do these cells respect the force balance? And you can do this in different tissues, and right, the way you would do this, you would define this compatibility variable, take the logarithm, plot the distribution. Of course, this data is noisy, so you don't expect to find the exact result, but you convince yourself that the tendency towards satisfying this constraint is so strongly statistically significant in some of the tissues that you look at. Not all, but often. Yes, please.
If you were going to look at a time course of these images, how much do you expect the angles to change over, say, minutes, or are they kind of stationary?
Thank you very much for this question. Please hold it for a few minutes, because we're going to turn to dynamics pretty fast.
What's your null
But
model?
yes, please.
Sorry, on the previous page, what is your null model?
Scramble. So we can scramble angles while we're preserving the distribution. It's all delicate, but there is, yeah. We're certainly comparing to the null. Okay. So the next thing you notice is that while the cell array, this tiling, defines a dual triangulation, the converse is not true. One can show that for geometries that allow triangulation, there is sort of a soft mode, 1 per cell, which corresponds to deformation that preserves angles. So kind of a discrete conformal symmetry there. So you basically expect that tissue in force balance then should also possess these soft modes, which are easily recognizable for, right, because they're orthogonal. It's just geometric criterion. We don't need to look inside. We just need to look at them. And again, we go to the data, and in that movie that I showed initially, right, one of the things that happened, well, always happens. One of the things that happens is, right, the tissue, right, its gastrulation process moves ventrally and is internalized, right? And just before the internalization, one sees a sort of very drastic deformation of cells, and then you can look at it more closely. And,
so this is folding, it's starting like this and folding inwards.
Yeah, so the mesoderm goes in. So there's a huge deformation, but before they start going in, there's just apical deformation, and that early deformation, curiously, is mostly isogonal. I just gave you little examples of how this instantaneous fast mechanical equilibrium constrains the geometry of cells, and how it is, can be verified and falsified. So, but in fact, one can do a bit more. This geometry is so constraining that we can actually use this as a tool to infer local tensions. So it takes a few minutes of counting, but you can convince yourself that local force balance constraint force balance constraints are enough to estimate local tensions and pressure. So in a way, one can read out the stress just by looking at the pictures. Fine. So we worked out some algorithm for doing the inverse. Then the question is, okay, how do we test this? Because there is no good, non-destructive way of measuring internal tensions. Well, so what we used is the fact that one can actually fluorescently label and visualize myosin. And we thought, okay, myosin is generating tension, so local myosin intensity should correlate with tension. And then we take a snapshot, and we do analysis. Madhav Mani was a postdoc at KTP a few years back. He's responsible for doing this, and then we can basically correlate predicted tension to observed myosin. And I'm plotting the cumulative distribution function. And again, as usual, I'm comparing so the inference result to control where everything is scalable, and the median correlation to control is 0. The median correlation in our analysis is 0.4. And now the question is 0.4 good, or is 0.4 embarrassing? Well, I would like
This is showing the amount of myosin along the edge,
yeah.
the inferred tension.
Yeah. So the analysis basically is done on kind of cell-by-cell basis. And so we have sort of low accuracy, but lots of points. And so then you just do it on the CDF. So I really don't understand what's going on, but I can't figure out. There it is. There it is. Now I can play a little movie. So I would basically argue that it's really pretty surprising that we get anything there at all. And now I come back to your question. The bloody thing is model static, right? It is dynamic. And the big question is, what am I doing here, standing in front of you and talking about force balance and the sort of very static configurations when we're just looking at static, like at snapshots of a dynamic process. So, so hold this thought for a moment. So we clearly have to go beyond the static picture and think more about dynamics. So I'm back to my little toy filament, and now I'm going to remind you that this wasn't supposed to be just a Hookean spring. It's an active element with myosin motors that can generate tension and do work and contract the filament. So they can start walking and shorten the filament if they can, and whether they can or cannot depend on the load of the filament, right? And if the load is too high, this filament will basically extend, so it will behave kind of as a plastic element. But if the filament is underloaded, it will contract. So that's what we expect. And with local behavior like that, you can convince yourself on in some continuum limit, you would just expect viscoelastic also, active viscoelastic behavior. So viscoelastic response forced by myosin-generated stresses. Well, so viscoelastic so it means that the tissue is a fluid, but that is a bit disturbing because you know there's a epithelium on my arm and it's not gripping. So it is somehow, it has shear rigidity, it is solid. Then you ask, okay, so how can this tension network actually ever reach mechanical equilibrium so that it can exist in that force balance? So remember that to achieve mechanical balance, tension has to triangulate the plane, but tensions are generated by myosins. So we need just the right amount of myosin on every edge so that we achieve the local balance. So now these myosins that are locally generated have to tile the plane. They have to be coordinated. We clearly need some sort of feedback. And you think for a moment, what would be the simplest feedback that would stabilize the system? And it's kind of obvious. What you need is some sort of dynamic recruitment where in response to stretching, you recruit more myosin, right? So if external tension is unsupported by your local amount of myosin, that's a signal to get more myosin. On the other hand, if the opposite is true, and chemical interface is generating too much tension to be supported by its environment, that's a signal to get rid of the mice. Okay? So, and you can show that this allows a Latournal functional and it is globally stable and is a well-behaved feedback. So the only problem is that we didn't know if any such thing actually existed in biology. We just thought that it would be good for the epithelial cells to engage in something like that. So it was a prediction, and then Sebastian Streikan and his students went to specifically look for some correlation between rate of strain and mice in recruitment, and indeed found that some such thing was there, although of course it's not linear, it saturates, and so on and so forth. But again, so a little illustration that some very simple and general ideas in this particular case, but so before we said, okay, fourth balance, right, should be relevant to biology. Stability should be relevant to biology. And these simple ideas turn out to be useful. So we end up with the following little picture. So we have 3 time scales. On the fast time scale, we have essentially just simple elasticity. Mechanical equilibrium is rapidly attained. Intermediate time scale, mice and activity kicks in, but also plasticity. So that on this time scale, we basically recover viscoelasticity, but I should have said here active viscoelasticity, because we do have internal myosin source of tension. But then there is yet another time scale. There is a slow time scale on which myosin is recruited and de-recruited from interfaces. So that's the time scale of dynamical feedback and biological control. And so in particular by this dynamic feedback construction, we expect that on this slower time scale, tissue can restore mechanical equilibrium and basically acquires shear rigidity. So I like to think of this as sort of an active solid, as opposed to an ordinary solid. And so the unusual property of the solid is that it actually flows on short times, but is rigid at long times. So I guess in my abstract, I alluded to something not in Landau and Lifshitz. So that's the kind of thing you wouldn't necessarily encounter until you start looking at something like a tissue that has so much activity and capacity for internal regulated modification. Yes?
Yeah, I just had a quick question about whether myosins are conserved within each of the cells.
I'm sorry?
Whether myosins are conserved within each of the cell over the slow time scale. Like, are there production of new myosin going on?
Well, so once we're into slow time scales, there are lots of slow time scales, right? So, there are many lines of defense. So myosin is transported from sort of inside the cell to the surface. Even on the surface, it can be assembled into filaments or not. It gets phosphorylated multiple times. There's a lot of regulation there. Yes?
For this slow time scale, what causes the extra strain, I guess? Like, I guess biologically, what causes the extra strain that causes the recruitment of extra myosin?
I'm not sure I entirely heard your question, but I'm going to guess. Do we understand the mechanism of the feedback?
No, but like the reason for it, like you said that it's because of extra strain, right? For a change in strain. So why does that happen? Is this like in reaction to growth? Is this in reaction to injury? Like,
well, so that's a question about the mechanism, right? So what I gave you is just a pure phenomenology. That you can characterize how much, the rate of strain, and you can observe the rate of accumulation of myosin on the surface, right? So that's just an empirical observation. Why and how that happens is a somewhat different question, and so the answer is I don't know. Very interested in understanding it, right? So can we actually dig deeper and uncover the mechanism that senses rate of strain? So we have some ideas what might do, but if you think for a moment how you would actually sense rate of strain, it's not entirely trivial. Yeah.
You mentioned pressure differentials early on, but it seems like then you drop them.
Yeah.
If you have this soft mode going in, you would think the pressure could build up and stabilize it. It sounds like you're saying that the actin growth or decrease is clarify.
Yeah. So I don't want to worry about it right now. We do worry about it, and it is relevant. And the soft mode, of course, isn't really that soft. Yeah, so you're right, one has to worry about it. So, let me go back to that example of this global flow in the embryo that Sebastian has provided us the data with. And now I can actually show this this thing moving, but never mind. So Sebastian can quantify the flow field as a function of time. We would really like to understand this global flow field. What can we do? So, we just talked a fair bit about myosin. Myosin generates local stresses, clearly relevant. Wouldn't it be nice if we could characterize the global distribution of myosin as a function of time? Again, Sebastian can do that. Myosin is localized in cell interfaces, and if you just stare at this picture for a moment, your eyes will pick up the local anisotropy in this myosin distribution. So we really want to quantify it as a tensor, and that's what we do. And then to describe the flow, we can take an active matter hydrodynamics approach and say, okay, so there's myosin, it generates stresses. The divergence of a stress tensor is local force, scale, we expect tissue to behave as a sort of viscoelastic material. So we're just going to model it as basically by Stokes equation with a couple effective viscosities. And basically we're going to take the measured Myosin stress tensor and solve for the velocity field, and then compare to Sebastian's measurements of the flow field. And the outcome is that...
can you go back?
Yeah.
So you're measuring the M and measuring the V's, and then you're asking for the consistency of this.
Yeah, so both sides of this particular equation is measured. Of course, we don't know the parameters, but our parameters are global. And the bottom line is that one can achieve a pretty good agreement with, and the truth is I don't know what's measured and what's predicted. Yeah, so measured is red, predicted is blue. This is the difference. There are some regions where comparison is not as good as we'd like, but the truth is I certainly did not expect that just with 3 global parameters one could actually describe global flow.
So what's the third one? You had nu and nu prime, and then this.
Well, that's hidden in Myosin tensor. So there. I'm bragging. One can predict something, but in reality, of course, we predicted very little, because all that's happening is we're looking at instantaneous Myosin field, and we're just computing velocity at the same moment. And what we really want to predict is what's happening in the next moment of time. And at the moment we have, like what I just showed you, we have no capacity for doing it. But we would really like to do that. So, but of course, in order to get there, well, we need to understand dynamics of myosin. Myosin is local property of cells. We need a description that sort of goes to the scale of cells, right? We're not going to get there in a continuum limit. So we're going to revisit the study of morphogenetic flow now with more imaging data and better imaging data, and now imaging data of such quality that one can get basically this polygonal tiling on the whole embryo, and track cells sort of from the beginning to the end. So these different color codes allow you to see where the cells are going. And this wasn't done by us. That was a heroic effort of Thomas Stern in Eric Wischhaus's lab. But the data is from Sebastian Streiken's lab. I am seriously running out of time, so I'm going to go through this very fast. So, it has been appreciated for a long time that if one looks at this process on the scale of cells, what one observes are two kinds of things. So cells deform, and cells intercalate. They change neighbors. And this is referred to as T1 transition, right? So topological transition, exchanging neighbors. And so the contributions of these two different modes of deformation are actually modulated, because so more intercalations happened in the lateral region, more deformation happening in the dorsal region. It's well, let's say fairly complicated. Two of my young collaborators, Fritjof Browns, who is a postdoc at KTP, and Nicholas Clausen, who is a graduate student, what they've done is sort of gone beyond just sort of classification of deformations of shapes, and they literally ran this tension inference algorithm that I I advertised earlier on this whole embryo as a function of time. So in principle, what they do is they basically assign an estimated tension to every edge, and they do it as a function of time. And what I'm going to draw your attention to is the following situation. So early on, we start with some sort of tiling, which is more or less hexagonal, so clearly disordered, but and it doesn't have any particular order in tensions. And but then one observes kind of the emergence of an alternating pattern of high and low tension. And the next thing that one observes is that high tension edges then proceed to contract and then generate these T1 transitions. And now the amusing aspect of the story is it turns out that the extent of order in the initial state is very important. Because if you look more carefully, you discover that there are sort of short range, but very significant correlations in hexatic order. And it is the presence of that order that that allows for an orderly pattern of tension modulation to emerge. And it is the destruction of this order that that arrests the elongation of the flow. So in a way, the extent of the flow was encoded in rather subtle order of cellular tiling at the onset. And anyway.
What happens to the cadherin when you have the interpolation?
Well, it interesting question. Well, it so the exact story is a little tricky, but all of these things are dynamic, right? So I think it's we should be thinking of both coherent and actomizing, basically constantly assembling and disassembling. And if you think that way, then there's plenty of time for it to re-equilibrate to a new topology. Okay, anyway, I should be wrapping up. Our current understanding of what's going on draws basically on this active solid picture. We can understand and model what's happening basically by thinking through remodeling of a force balance. So so the whole story is about recruiting myosin, and in addition to what I told you, in order to make this happen, we have to postulate positive feedback. We need high tension interfaces getting stronger. So that's not a prediction. We haven't tested it yet. But with that additional ingredient, Fritjof and Nicholas can put together a model which reproduces this global behavior quite well. So again, the assumptions are rather simple, but there are falsifiable predictions. And one of the interesting predictions is exactly the relation of this sort of global control of the flow by the ordering of cell lattice. And there are mutants that generate more or less ordered cell packings, and there are certain processes that we know are involved in generating more or less ordered packings, and this will all be tested. So, I guess my final message is all that I described to you was phenomenological, and just trying to identify a few general, simple physical principles, and follow it through to falsifiable conclusions. And I would argue that it has served us pretty well, and will serve other people too. It generates hypotheses, it provides useful tools, and sometimes we learn new things about physics. And my other message is maybe biology isn't as complex as we usually think. So I'm very optimistic for this, rather than sort of worrying about emergent complexity. I'm all for emergent simplicity. I think I'm going to skip this straight to the acknowledgements. So I pretty much, I think, mentioned the key players. Fritjof, Brown's a postdoc at KTP. Nicholas is still a student at UCSB. Dylan was a student at UCSB, now is a postdoc at Rockefeller. Nick Knoll is actually at least working in Palo Alto, although I don't think he's living in Palo Alto. And Madav and Itzia, former postdocs, who now have tenured faculty positions. Sebastian Strekan was the source of all the data that I showed, and have done some of the key work. And we've also enjoyed wonderful collaboration with Eric Buschhaus and his friends. Thank you very much for listening.
Maybe we have time
Yeah.
for one or two.
Let's see. So, Boris, very nice talk. Now, I have a big question for you.
Yes.
Having to do with this overall structure of the network. And it's a question about the control machinery that organizes the unfolding. Now, you've got two choices. One is the cells remember. And they have a program that they execute. And I think the other is that there's some sort of guiding field. I can't think of myself of any other options. Now, do you have any ideas about what is coordinating the ramping up and down of the stresses in various cells?
I think about it day and night. I think the history of the subject has gone, right, so Turing's idea was kind of bottom-up self-organization, an instability generating a pattern. Then so geneticists discovered that the stripy pattern that I showed somehow, instead of being self-organized, is so stupidly genetically controlled one stripe at a time, so you can just remove two stripes in the middle, and now it's so supposed to be sort of a counterexample that has sunk Turing forever. I think what we've learned is that that's not so, that it's sort of an exception rather than the rule. That fly is a particularly big hurry to do it. So, and the truth is probably somewhere in between. That so I should have said that of course these pattern of stripes emerges in response to what's called morphogen gradients, so I can tell you. So a graded distribution of some factor which controls gene expression concentration dependent way. So this kind of top-down patterning, right? So my bet goes on so two mechanisms working hand in hand, right? You can't leave something like patterning of the embryo to spontaneous symmetry breaking. But on the other hand, you can't pattern the whole thing just by measuring or reading out gradients of something. So these sort of large-scale morphogen gradients are like fields biasing for the symmetry breaking patterning generated bottom-up. And I think so a number of examples towards this interplay of bottom-up self-organization and top-down control is growing. Sorry, I think you've always...
Yeah, I'm just curious, I'm not sure how relevant this is, but do these dynamical equations admit sort of periodic solutions, like there are various periodic processes in organisms? I don't know if that matters.
So they're definitely, they're famous cycle, right? The cell cycle. There is yeah. So most obviously in the context of stuff that So I was showing this striping pattern of gene expression. One way of generating it would be so one stripe at a time. This is how this process of mutagenesis, right? This is how vertebrates grow their spines. So it is a periodic process.
But I mean, ultimately, like the heartbeat or the
yeah, sure. There's all that.
how the global, essentially the global geometry of the embryo plays into all these, essentially into, let's say, determining the initial packing, right, of the lateral, into the convergent extension. So like, yeah, hold on a second. In this, yeah, in the back, yeah. So that if these movements are so sensitive, let's say, to the fine structures of the packing, then those packing must be somehow then, like that, the correlation structure must be preserved, you know, in embryos of, let's say, varying lengths and varying curvatures and so on. But is that true, or can you use also mutants, let's say, of the global shape of the thing, like a more round, like the ball shape, or?
So let me. You're asking a complex question, and I'm going to pick an easy part of it to answer. Yeah. That's okay. So one thing that I didn't mention about where we think this pattern of packing is coming from is basically a hot dog effect. There is anisotropic stress due to tubular pressure in the embryo, right? So, right, azimuthal interfaces are so preferentially loaded, and it's the feedback driven by tubular pressure that the thing kickstarts the whole process. This is controversial, and I know my talk is being recorded. So if Eric Bushhouse, hears me saying this, I'm in deep trouble. So that's the elementary example where we believe the shape of the bloody embryo is crucially important. Generally, the question of how patterning depends on morphology is super interesting. So with organoids, right? So people start growing stem cells in the dish, they form little shapes, and the question is, okay, so there are these morphogens diffusing, but now they're diffusing on surfaces, the shape of which is controlled by, say, morphogens that I think what we're learning is that it's not just genes, it's also shapes, morphology. You have to pay attention to this. So, yeah, interesting question.
Maybe last question, and we'll get some more questions.
So when you said the Navier-Stokes effect, I understood you right, they don't really work.
Just Stokes.
So can't you use the measured myosin as an input to get the velocity fields? And if you do that, so is your statement, I'm just trying to understand what you're saying. So is your statement that if you do that, you get the wrong velocity fields? Or
No, you get the right velocity field. Well, again, sorry, I lost that particular thing. What I was trying to convey is that this tissue is both fluid and solid. And you can describe it, well, equally well, I don't know how equally it is because you're describing some of the different aspects of it, but both points of view are useful. Okay? And it's rather subtle because the way it flows is by adiabatic rearrangement of force balance. Right? It's not, but you can also correlate that with imbalance in myosin distribution. And you can, well, anyway, I can talk about it for hours. You don't want that.
Okay. Well, let's thank Boris one more time.